Welcome back to my channel Knowledge Chronicle.
Have you ever wondered what makes you you? From your eye color to why some medicines work for you and not for your friend. Today, we're diving into the Human Genome Project, the giant scientific mission that read the instruction manual of life. Stay with me. By the end, you'll understand what they did, how they did it, what they found, and why your future doctor might one day read your genome like a lab report. Quick fact: sequencing the entire human genome was once thought impossible. Now personal genomes cost a few $100s. That's the revolution we'll explain, simply and clearly. Aim and duration of the project. In plain words, the Human Genome Project, or HGP, was an international effort started in 1990 with one main aim: to identify and map every base pair in human DNA and to find all human genes. So, let's break down some important terms. DNA bases, or base pairs, are the fundamental units of our genetic code. DNA is made up of 4 chemical letters: A, T, C, and G. 2 complementary letters pair up to form a base pair. The human genome contains about 3 billion base pairs. That's the text that needs to be read. Now, a gene is a stretch of DNA that typically gives instructions to make a protein. Early on, scientists expected there would be tens or even hundreds of thousands of genes. But the Human Genome Project revealed that humans actually have roughly 20,000 to 25,000 genes. The project officially ran from 1990 to 2003, with a working draft announced in 2000. So that's about 13 years of coordinated laboratory work, computing, and international collaboration. So, how did they actually read DNA? Well, they used several strategies and tools. You can think of it kind of like turning a shredded book back into readable pages. One key method was Sanger sequencing, which was the original and highly accurate technique used extensively by the Human Genome Project. Imagine copying a sentence, but stopping at random letters to see how long the fragments are. By looking at many fragments, you can reconstruct the whole sentence. Sanger sequencing is slow, but it's really accurate. Then there was map-based, or clone-by-clone sequencing. Here, scientists broke the genome into large, ordered pieces, kind of like chapters in a book, mapped them to know their order, and then sequenced each piece. This approach helps reduce confusion when assembling repetitive regions. Whole genome shotgun sequencing was another method used by private groups like Celera. In this strategy, you shred the whole book into tiny pieces, sequence everything, and then use powerful computers to assemble overlapping pieces into the full text. This method is faster, but it really needs strong computational assembly. After sequencing, computers align overlapping fragments into what are called contigs, or continuous sequences, and then into chromosomes. This step is like solving a giant jigsaw puzzle using billions of tiny pieces. Later on, next-generation sequencing, or NGS, made sequencing massively faster and cheaper. The Human Genome Project relied on older methods, but it really set the stage for NGS. What was discovered? The surprises. The Human Genome Project gave us hard numbers and, honestly, several shocks. The gene count was small, only about 20,000 genes, which was far fewer than anyone expected. That really forced a rethink. Complexity doesn't come just from the number of genes. We also learned that repetitive DNA makes up much of our genome, and it's not protein coding. There are these long stretches of repeats and mobile elements, sometimes called jumping genes, that actually influence how genes are regulated. Another big finding was the cataloging of millions of single-letter variations between humans, called single nucleotide polymorphisms, or SNPs. These SNPs are really the basis for genetic differences and for studies on disease risk. And we found out humans share about 98 to 99% of our DNA with chimpanzees. But those small differences can have huge effects. For example, discovering BRCA1 and BRCA2 mutations as strong breast and ovarian cancer risk factors came directly from gene mapping approaches that the Human Genome Project really accelerated. How it changed modern medicine and genetics. Real-world effects. The Human Genome Project moved genetics from being just a curiosity to a real clinical tool. Some key impacts include disease gene discovery. The project created reference maps that let researchers find the genes behind inherited disorders quickly. A good example is diagnosing rare genetic disorders using sequencing. In cancer genomics, instead of treating all cancers the same, we now sequence tumor DNA to find targetable mutations, like HR2 in breast cancer, which can be targeted by drugs such as trastuzumab. Pharmacogenomics is another area. It means predicting drug response from your genes. For example, variants in CYP enzymes affect how you metabolize things like warfarin or antidepressants, so dosing can be personalized. Genetic testing and newborn screening have also become faster and more accurate for inherited conditions. And there's been a biotech boom. Vaccines, diagnostics, gene therapies, and even CRISPR, which came later but totally builds on genome knowledge. But not everything is solved. Many common diseases are polygenic, meaning many genes plus the environment are involved. So genome information improves risk estimates but rarely gives absolute answers. What's next? Personal genome mapping. The future and cautions. Personal genome mapping means sequencing an individual's genome to actually guide their medical care. So, what can you expect? We're looking at actionable medicine. Things like tailored drug choices and early screening for higher-risk individuals. For example, BRCA carriers can receive enhanced surveillance. Polygenic risk scores are another thing. They combine many small genetic effects to estimate disease risk, but these are still imperfect, and population bias is definitely a concern. Then there are the ethics and privacy issues. Your genome is permanent and identifying. Who owns it? Can insurers discriminate? Laws exist, but they vary, and privacy is a major issue. Data interpretation is another challenge. Sequencing is cheap now, but figuring out what all those variants mean is the hard part. Many variants are still variants of uncertain significance. And just to be clear, two people can have the same risk variant but never develop the disease, because environment and lifestyle matter. Genes are not destiny. So that's the Human Genome Project. A 13-year global effort that read the instruction book of life, revealed surprising biology, and powered a revolution in medicine. From diagnosing rare diseases to guiding cancer therapy and launching personal genomics. The science is moving fast, but the real challenge now is using genomes wisely, accurate interpretation, ethical safeguards, and equitable access. Your one like, share, and subscribe will help us create more valuable content.